methods to control these microbes, right? All these methods are like applied at different places. First, we'll talk about physical methods for controlling these microorganisms, right? Before that, we will talk about what a disinfectant. All of you must have heard this terminology, disinfection and disinfectant. Disinfectant is any substance or is usually a chemical that destroys disease-causing pathogens or microbes or other harmful microorganisms, but might not kill bacterial spores. That means they are able to kill the vegetative spores, but they are unable to kill this bacterial spores, right? They refers to the substances applied to inanimate objects. That means they can be applied only to the non-living. They cannot be applied. Have you ever heard that disinfectant is being applied on skin? No. These are like we are using ethanol or any sterilizer, right? But we are not using disinfectant. We are using disinfectant on inanimate like fridges, tabletops, right? Okay. So disinfectant is applied on inanimate objects. Then there is disinfection, the process of disinfecting, right? These are less lethal than sterilization. Sterilization, it means different, right? Sterilize to complete eradicate the microbes, right, from any surface or from any particular, what do you call, object, right? That is 100% sterilization removal. But in case it is not disinfection, it destroys most recognized pathogenic microorganisms, not necessarily all microbes. Okay, so disinfect. What is the why it is required to prevent the transmission of certain microorganisms with objects, hands, or skin, or prevent spreading the infection, right? Okay, as I've told you, disinfectants could be based on their what do you call level of effectiveness. They can be categorized into three types. One, high level disinfectants, right? They are active against bacterial spores also, right? Then there is intermediate level. They are effective against tuberculosis bacteria and but not against spores, right? Then there is low level disinfectant. There are wide range of activity against microbes but not against spores and tuberculosis, right? They'll cover most of the microbes, but excluding these two, spores and this one, tuberculosis bacteria. So chemical methods there are different chemicals, right, which can be used for killing or what do you call removing or inhibiting microbes, right? So mainly there are two principles by which we can reduce or eliminate microbes, right? One is inhibiting to like whatever number, like we are applying these chemicals and suppose Mm, yes, if suppose they are being used, any certain chemical is applied and they are not able to kill 100% bacteria, but what will happen? The amount of what do you call microorganism, they will remain as a static. That means number is there, they will be there, they will not multiply. One is that. Another one is killing or cytal effect, right? That means microbes can be controlled by two methods. One by killing the microbes, another one by inhibiting the microbes, right? So among chemicals, first one is like there are certain disinfectants. We have seen what are disinfectants and disinfection, right? There are various types of disinfectants. For instance, first one is phenol and phenolics. Phenol, we all know this was the most, what do you call, oldest, right? For the first time, it was the first antibiotic. It was discovered by Joseph Lister, right? Lister, Joseph Lister. It was discovered by this one. Okay, and then what are phenolics? The derivatives of phenol, right, is known as phenolics. So phenol, which is generally carbolic acid, was first used by Joseph Lister as a disinfectant, right? It is rarely used today because phenol is like irritant and it is bit carcinogenic also. And also a strong odor. It has very pungent odor and it is bit carcinogenic. It is used in some throat sprays and lozenges. Acts as a local anesthetic. It can be used for anesthesia purposes. Then there is phenol related compounds. We have already seen like what are intermediate level, high level and low level, right? So these phenols, they come under internal level of disinfectants, right? So phenols are obtained by distillation of what do you call in this one? Petroleum, coal tar. That is distillation of coal tar between temperatures near 170 and 270 degrees Celsius, right? So they are obtained from there. If 1% phenol is bactericidal, which denatures how they act, what is the mode of action, what they do. Phenol mode of action, right? Okay, what they have OH group, right, carbon. So this one, they will react with the proteins or enzymes, this one OH, and they will affect their activity. Like suppose any enzyme which is being used for application or other like normal activity, it will be inhibited, right? So which denatures protein, right? It will be denatured and disrupts the cell membrane. Cell membrane phenol, they will come over here and they will denature, disrupt the denature the protein and they will disintegrate the cell membrane, right? Effective in the presence of an organic matter and remain effective for a prolonged time. They can be, if organic matter is there, then they'll become more effective and it could be there for a longer period of time. Generally, it is used for in healthcare like hospitals, labs, and homes. Lysol and tricolozone is commonly used. Their disadvantages, it is an irritant and it is cancerous also, have disagreeable odor and possible side effects. It is rarely used as an antiseptic or disinfectant because it irritates them and has a disagreeable odor. Right. Then what are phenolics? These are the derivatives of phenol and are called phenolics, right? Phenolics exert antimicrobial activity by injuring lipid containing plasma membrane, right? How do they act? By they injure the lipid containing plasma membrane, right? Which results in the leakage of cellular content. So all the cytoplasmic content, they will come out. Okay. Phenol, which is again carbonyl, not effective again, not ineffective as a disinfectant. There is a crude phenol, phenol and cresol, effective against gram-positive and gram-negative organisms, right? One is that was phenolic. Another one is cresol, right? Okay, they are also from like from phenolics only. Okay, cresol, they are derived from the coal tar lysol, right? Okay, bipheноls, or it is sold in the market by this name, Physohex, effective against gram-positive and like Staphylococci, it will be capital Staphylococci and Streptococci, right? Used in nurseries and excessive use in infants may cause neurological damage, right? Nurseries where this one immature babies are kept. Okay, destroy plasma membrane and denature proteins. Cresol emulsions, very powerful disinfectant. Advantages, they are stable, persist for a longer time after application and remains active in the presence of organic compound. Where they are used, uses in hospital, they are used in hospital. 5 to 15% is used for disinfection of urine and fecal matter. They are active against gram-positive and gram-negative bacteria and moderately against Mycobacterium tuberculosis, Mycobacterium, and have ability against spores and viruses, right? Okay, fine. Then come to the another chemical which is commonly used as a disinfectant. One these are halogens. Halo, salt periodic table. That these one like chlorine, fluorine, bromine. This group, right? So halogens, right? Okay. So like there are certain salts and there is chlorine commonly used. Then there is the different forms of iodine is also you commonly used, right? So halogens, they are effective alone or in compound. Like They will be alone will be effective, or they are mixed with certain organic compound, right, to increase their effectivity. So they inactivate. How they do act? See this one. They again inactivates the enzyme by converting functional SH group to this group is being converted into halogens. What they do into SS oxidizes, right? So activity of that enzyme will be lost to the oxidizes SS form. For one is iodine, tincture of iodine, which is commonly used, right, from the ancient time. Alcohol, alcoholic solution, and one of the first antiseptic used, right? It combines with amino acid tyrosine in protein and denatures protein. How do they work? They combines with the tyrosine, right? This one, halogen, if you will say iodine, iodine reacts with tyrosine, which is present in protein and denatures them, right? They denatures them. Okay. Then there is iodophores, iodo compounds with iodine that are slow releasing. They releases this one iodine very slowly and take several minutes to act, right? They'll have sustained but longer effect. Used as skin antiseptics in surgery, not effective against bacterial endospores. They can't be used against endospores, but they are quite effective against vegetative spores. Then there is chlorine. Chlorine, these are commonly used in drinking water, right? So these are being used. So what is there? How do they work? They're like any other chlorine or they're any other salt, right? This chlorine reacts with the water to form hypochlorous acid. Hypochlorous acid, HOCl, right? Okay, this one, HOCl, sorry, they form HOCl, hypochloric acid. Okay, so in addition to chlorine itself, there are three different types of chlorine compounds. One is hypochlorites, right? Inorganic chloramines. Inorganic compound will be there with chlorine, and there is organic chloramines, right? It is also commonly used. The disinfectant action of all chlorine compound is due to the liberation of free chlorine. All these, what you call their compounds, they will release chlorine. Chlorine, which is the main disinfectant, right? Or which have the more and which has the what do you call as a major disinfecting agent, right? So mode of action: when elemental chlorine or hypochlorites are added to the water, chlorine reacts with the water to form hypochlorous acid HOCl, which in turn neutralizes or acidic solution is a strong oxidizing agent and an effective disinfectant. So this is what happens. Chlorine reacts with the water. They will liberate this one. It will get free, and this one there will be formation of HOCl. This one is the main what you call disinfectant, right? It is used to disinfect drinking water, pools where commonly like swimming pool. If suppose any patient, anyone is contaminated, anyone is having infection of certain what do you call pathogen, and he or she is what do you call shedding continuously shedding this pathogen into the swimming pool, and he like we can say they are carriers, right? If another normal healthy person will come to those contaminated water, there's a possibility of getting infection. So this one, it is advisable to what you call treat all such swimming pools where it is accessible to most of the common people, right? And sewage, chlorine is essentially inactivated by organic materials. Sodium hypochlorite, NaOCl, that is NaOCl, is an active ingredient of bleaching agent, right? Chloramines consist of chlorine plus ammonia, less effective as germicide. Germicide, germs. They are used for less effective as a germicidal to kill the germs, right? Okay. This is how like chlorine acts on the. This is normal bacterial cell. This plasma membrane. HOCl. We have seen there is a formation of HOCl over here, right? This one. So what will happen? This one, they will enter over here, H will be released, and this one, chloride. And they will attack and penetrate the cell wall and membrane and enters the cytoplasm, right? Okay. Then they will damage this one, what do you call, the major machinery over there. They will cross this barrier, they will disrupt this one, right? They will penetrate, okay. All the cellular components will be, once they will damage, all the cellular contents, they will come out. Ultimately, the bacteria is killed, right? The same process, right? Okay. Same thing is given over here. There is a pathogen or mini microbe. This is we have studied that they have a certain charge, negative charge, right? Okay. Okay. Some bacteria will have positive charge also. This one they will enter and they will, what do you call, damage the cytoplasmic content. They will come out, right? Okay. So this one is the main agent for killing or this what do you call, affecting these microbes. Then there is ethanols or alcohols, right? They have OH group. They could act as an acid, right? OH group. So what happens? They in this one, what is the mode of action? They interacts with the proteins, that is against proteins or enzymes, and makes them inactive, right? They denatures the protein, dissolves the lipid. Yes, it is they are organic solvent, right? So lipids will get easily dissolved or disintegrated. As we have seen in case of gram-negative bacteria, they have more lipopolysaccharide, this one, lipids, so it will get dissolved and can lead to the cell membrane disintegration. Cell membrane will get disintegrated. They kill bacteria and fungi, but not endospores or naked viruses. But they are not effective against endospores or naked viruses. Used to mechanically wipe on the surfaces, microbe skin. Generally, you must have noticed when during injections, the skin is being sterilized first, or this one. It is being wiped with ethanol, right, to remove the pathogen microbes from that surface. Otherwise, they will enter to our, what do you call, blood, microbes of the skin before injecting or blood draws, right? Not good for open wounds. We can't use this directly for the open wounds because cause proteins to coagulate, right? They will come into the cells and they will coagulate, affect the tissues, right? Ethanol, most commonly used is isopropyl alcohol, 70%, right? Most effective are the most frequently used. Isopropanol rubbing, right? Okay. Better disinfectant than ethanol, this one, right? Okay, 100% is not that much effective in comparison to this one. Also cleans and it is comparatively, if we using this one 30%, that means we are using 30% water. And at 30% water, they are cheaper, right? So the final formulation is quite cheaper. And our ethanol is volatile, right? So if we will add water to this, there'll be their volatile nature will be decreased. So that means this is the benefit, right? Then there are heavy metals. We must have seen there are various heavy metals like copper, lead, nickel, zinc, gold, silver. All they, how do they affect the cells? How do they affect the cells? Again, the ions produced from these heavy metals, they interact with the, what do you call, again, proteins or enzymes, and they'll make them inactive, right? Ions of heavy metals such as mercury, silver, arsenic, zinc, selenium, copper are widely used. Ions are antimicrobial because they alter the 3D shape of proteins. How? That is, they changes the conformational structure, right? You must have seen that there is supercoiling, conformational 2D structure, right? So it is being altered, inhibiting or eliminating their function. Heavy metals combined with the proteins, I have told you, heavy metals, any heavy, suppose Cu, it will react with the proteins. They have different amino acids, right? Proteins inhibiting enzyme activity, so activity of the enzyme will be inactivated, right? Affected, often with the sulfhydryl group. I have told you here, this group, SH group is affected, right? They'll become ineffective, right? Mm, and inactivate them. They may also precipitate cell proteins. The protein they will interact these heavy metals, suppose Cu. Cu will react with any this one proteins and they will precipitate them right within the cells. So this is how they affect, right? Oligodynamic, that means whenever they come in with this one organic molecule, they are quite effective. Very tiny amounts are effective, right? Okay, small amount is required. Heavy metals. Silver is widely used. We have seen like we used to this one, what do you call? Silver vessels, silver pots, right? That was one of the main reason we used to have copper vessels, copper pots. We used to drink water. That is again because of this same reason, right? Okay, the most commonly employed of the silver salt is silver nitrate is widely used, right? 1% silver nitrate is used to protect infants against gonorrheal eye infections. Gonorrhea, it causes sexually transmitted dysentery, gonorrhea, right? And there is, and it is also present in this one, nasopharynx. So in infants, these are used, and it is also suppose somehow they are carried to eyes, they will affect the eyes. So it is being used as for this purpose, right? Then there is mercury. Mercury is like very harmful, what do you call, toxic. Organic mercury compounds like merthiolate and mercurochrome are used to disinfect skin wounds, right? It is very toxic and present as limited use, right? Mercury compounds in the form of organic derivatives, example, phenyl mercuric nitrate, acetate, thiomersal, and mercurochrome are less toxic. Mercury itself is very toxic, but when they are clubbed with these things, organic compounds, their toxicity is reduced, right? Are used as a then they could be used as antiseptics, right? They are active against both gram-positive as well as gram-negative bacteria. Then there are various heavy metals, right? Copper is also there, commonly used. Okay, copper, we used to have copper utensils. Copper sulfate is used to kill algae in pools, swimming pools, right? Okay, fish tanks. Selenium, it it is also used as antifungal. It has also antifungal activity, so it is used in certain shampoos, right? Dandruff-based shampoos kills fungi and their spores, right? And used for fungal infections, also used in dandruffs and shampoos, right? Dandruff shampoos. Zinc chloride is used in mouthwashes, right? Then there is quaternary ammonium compound, quartz. They are also called quartz. Quaternary ammonium compounds. They are the cationic, that means they are the positively charged, right? Detergents that are effective against gram positive and comparatively less effective against gram negative. Why? Because charge, they will have certain effect on cell wall, right? And also used to destroy fungi, amoeba, and enveloped viruses, right? But they are only less effective against gram negative. Widely used surface active agents, Zephyrin and Cipacol found in lab spray bottles, cetrimide or citavolone, better they have bactericidal activity against gram-positive organisms, but lesser activity against gram-negative. 1 to 2% is commonly used, right? So Savlon, you must have heard, combination of cetavlon and hibitane is used. Plus, plastic appliances can be disinfected within 20 minutes. Suppose any plastic surface is there. So if this will be applied, it will get disinfected in minutes, right? Disinfection of clinical thermometers, like we can't use, if suppose any patient is infected, we can't use the same thermometer to everyone, right? In that case, it has to be disinfected by this one, quartz, in within 3 minutes or Savlon, right? Advantages is that they are strong antimicrobial, they have strong antimicrobial action, colorless, odorless, tasteless, and stable and non-toxic, right? Disadvantage, since they are surfactant, they forms foam, right? Organic matter interferes with the effectiveness and they are being neutralized by soaps and anionic detergents since they are, right, surfactants. So their activity is affected. Then there are aldehydes. Aldehydes, okay, this group. Okay, aldehydes, they adds alkyl group, right? The lethal effect of aldehyde, formaldehyde, glutaraldehyde is commonly used, result from their alkylating action on proteins. So what happens? there is a protein, these aldehydes, they will add this three group CS3, C2H5, any group, and their activity will be affected, right? One commonly used aldehyde is formaldehyde. Commercially available, 37% formaldehyde. It is in formula for preservative, right? Or as paraformaldehyde. Active against the amino group in the protein molecules. It is lethal to bacteria and certain their spores, but less than glutaraldehyde, many viruses and fungi. It is employed in the liquid and vapor states. Generally, 2 to 3% solution is used for spraying rooms, walls, and furniture. This one, aldehyde, formaldehyde is used. Active at a high temperature and relative humidity of 80 to 90. Whenever there is a moisture humidity, their effectiveness will increase. Their mode of action, it combines readily with the proteins and is less effective than in the presence of organic matter. Denatures proteins and inactivates nucleic acid. There is another aldehyde, glutaraldehyde, which is commonly used after formaldehyde. They are less irritating and more effective than formaldehyde. One of the few chemicals that disinfects, that is the sterilizing agent. It can be also used as a sterilizing agent. A 2% solution of glutaraldehyde, which is known as Redax, is a bactericidal, tuberculosidal, and virucidal. Within 10 minutes, it will have all these three activated, right? Okay, sporicidal, it will take 3 to 10 hours, right? But most of the they are effective against viruses, tuberculosis, bacteria, and bacteria within 10 minutes, right? They're commonly used to disinfect hospitals, instruments, right? And also used in mortuaries for ambling, preserving mortuaries, dead bodies, right? Gaseous sterilizers, there are certain gases which are used for sterilizing purposes, right? One is ethylene oxide. What they will do, again, they will also add groups, ethylene group, right? They will add this group to any proteins or right, and they will affect their activity. Chemicals that sterilize in a chamber similar to an autoclave denatures the proteins by replacing functional group. Suppose any functional group OH is there, it will be replaced by this one with alkyl group. So the activity will be lost. Any enzyme, replicating enzyme is there, right? Okay. Colorless, highly inflammable. If you talk about, and they are highly colorless, highly inflammable liquid with boiling point of 10.7 degrees Celsius and is highly penetrating. They can penetrate what you call easily with a sweet ethereal smell by mixing it with inert gases such as carbon dioxide or nitrogen. Sorry, to a concentration of 10%. Its explosive tendency is eliminated, right? At this degree Celsius, it kills bacteria, spores, and viruses. Water vapor is added to the mixture to increase its efficacy. They are commonly used with water vapors to increase their efficacy, right? They are also used to sterilize fabrics, plastics, equipments, cardiac catheters. Peroxisons, they are oxidizing agent. What did they do? Like oxidizes cellular components of treated microbes, disrupt the membrane and proteins. One commonly used peroxisons is ozone, right? Used along with chlorine to disinfect the water. It is also known as ozonization. Helps neutralizes unpleasant taste and odors, more effective killing agent than chlorine, but less stable and more expensive, right? It is quite expensive. It is just packaged water bottles. This technique is commonly applied. Highly reactive form of oxygen made by exposing oxygen to electricity or by UV light. Then there are peroxides also. Hydrogen peroxide, it is used as an antiseptic. It is not good for open wounds because quickly broken down by, we have certain body have this one catalase enzyme. So the moment hydrogen peroxide comes in contact with the catalase, they form water, right? Present. So it is ineffective. In disinfection of inanimate objects, it can be used against inanimate objects. Sporicidal at high temperatures and used by the food industry to disinfect contact lenses. Thank you.